Dear audience, my name is Marcel and I'm very happy to present you today our work based on the topic of FUS-ALS. As you certainly know, mutations in fusin sarcoma are found in up to 5% of all genetic ALS cases and are mainly located in the C-terminal domain, which is coding for the nuclear localization sequence. Such mutations lead to an imbalance and disturbance of the physiological nucleocytoplasmatic ratio of the FUS protein. However, how this leads to motor neuron degeneration is currently not known. Therefore, we aim to establish a human, patient, and iPSC-derived neural system in vitro to study pathology FUS-ALS. To study individual axons, we seeded the neurons into tiny microfluidic chambers that allowed the guided growth of axons without contamination of any neuronal cell bodies. This resulted in the establishment of a pure distal axonal model system. This way we saw a gradual loss of distal axons only in the mutant condition over time, which was followed by neuronal apoptosis much later on. Interestingly, we saw signs of increased DNA damage in histology of affected patients. We also found evidence for this in our cell culture system. For example, adipocyte could induce even more DNA damage, but it also led to distal axonal phenotype, which was characterized by a loss of mitochondrial membrane potential of these mitochondria. The same was true for FUS mutant axons, but without any treatment, just under standard conditions. By using laser ablation microscopy, we were able to dynamically monitor the response of FUS to control the DNA damage. FUS wild-type GFP gets recruited to the site of DNA damage in a matter of seconds. However, this was not the case in the mutant condition, where we find evidence of mutant FUS GFP in the whole cytoplasm. By interfering with the relevant pathways FUS is involved in terms of DNA damage, we were able to restore this pathological condition. Indeed, inhibition of both enzymes PARC and DNA PK led to a rescue of the recruitment of FUS to the site of DNA damage in the mutant condition. Indeed, inhibition of the very same enzymes also restored the pathological condition in the distal axons of FUS mutant spinal motor neurons. Next, we were wondering if these signs of DNA damage might also relate to an increased cancer burden in affected patients, which was however not the case in our multi-center cohort study, where only 1 out of 36 novel FUS-ALS patients had a cancer history. Thank you very much for your attention, and in case of any questions, just send me an email or check out our website.